To the PSOM Cell and Tissue Mechanics seminar series. This month, March, it is my pleasure to introduce Professor Stephanie Fraley, Associate Professor of Bioengineering at the University of California, San Diego. She earned her bachelor's in chemical engineering at the University of Tennessee and her PhD at the Johns Hopkins University. She has contributed fundamental knowledge in the fields of 3D cell migration and molecular detection technology. And she has received numerous awards, including the Biomedical Engineering Society Rising Star in Cellular and Molecular Bioengineering, Biomaterial Science Emerging Investigator, and NSF Career Award. Without any further ado, the floor is yours.
Great. Thanks so much for the invitation, and I'm excited to talk to everyone. If you have any questions, feel free to stop me, in the middle of the talk. You don't have to wait till the end. Look forward to good discussion. Okay. Well, now I can't change slides. Okay. All right. So the main motivation that my lab follows is trying to understand and engineer cancer cell migration. And the reason is, you all know, dysregulated migration is the cause of cancer deaths. If you catch a tumor before it started migrating through the basement membrane and into the collagen-rich interstitial stroma, then you have a much greater chance of survival. If a tumor starts to break through and migrate away, this is really the cause of metastasis, which is what results in 90% of cancer-related deaths. And in my lab, we really try and take a systems engineering approach to understand cancer cell migration. So the goal is to predict and design migration outcomes, right? Be able to stop migration patterns that lead to metastasis. And to do this, we really believe in systems engineering, which is looking at how parts within the whole interrelate rather than the more reductionist view of looking at one individual part and its influence. And so we're not only thinking about adhesion proteins, cytoskeletal proteins, but we're also interested in thinking about how boundary conditions like ECM composition and mechanics and neighboring cells affect migration outcomes. And then, like good engineers, we have to be concerned with initial conditions and how these influence cell states and how plastic cell states can be. And hopefully the result of all of this work is, integrative models that can bridge across conditions, scales, and time. Within this framework, we apply systems engineering to two challenges that I'm going to talk to you about today. The first challenge is understanding migration heterogeneity across environments. And this is really talking about boundary conditions. So if you study a cell in 2D, which we've done for a long time, and we understand a lot about how cells move in 2D systems, a big challenge comes when you take that same cell out of that 2D system and put it into another system. So for example, here the cell is moving on 2D collagen. If we put it inside of a 3D collagen matrix, we get very different migration behavior. And over time, my lab and other labs have shown that proteins can localize differently, they can function differently, and cells can migrate very differently, depending on the microenvironmental context. So how is it that cells are sensing this change from 2D to 3D? How do cells sense dimensionality? That's one of the challenges I'll tell you about today. The second challenge is understanding migration heterogeneity among cells of the same type. And this is really focusing in on initial conditions. So if you put cells, for example, into a 3D collagen matrix, and these are MDA, MB231 breast cancer cells, you can see that some cells will spread out, invade collectively, and proliferate, while another subpopulation of cells will undergo rotational migration and proliferation. And so how is it that you can get two very different migration outcomes that lead to two very different structural cell structures with the same cell type in the same matrix. And how is it that you get different proportions of them as well? So we get like 86% of these that are forming these invasive network structures, and about 14% that are forming acini or spheroids. Okay, so the first part of my talk, I'm going to focus in on heterogeneity across environments, and I'm going to tell you two stories from my lab. The first one is the question, how does ECM dimensionality influence cell state? And the second one, we ask, what is a relevant ECM context in which to study breast cancer migration? And then in part two, we're going to talk about heterogeneity among cells. And we're going to ask, how do cells of the same type in the same ECM condition achieve these distinct migration behaviors? Okay, so part 1A, how does ECM dimensionality influence cell state? Well, we know from Dennis Disher and Adam's work, hi guys, that ECM stiffness plays a very large role in changing cell state. And from the Piccolo lab primarily, we know that YAP-TAZ localization within the cell is one of many different mediators of this mechanotransduction cascade from stiffness to cell state. And in 2D, how this works is that on soft substrates, very reliably, you get little mechanotransduction. And so you get, little to no nuclear translocation and transcriptional activity of YAP. But on stiff substrates, you get higher nuclear localization and transcriptional activity from YAP and TAZ. But what's interesting is over time, when folks have tried to look at 3D culture and see what happens, a bunch of different things happen. So sometimes folks have reported that cells do the same thing they do in 2D, where YAP goes to the nucleus upon stiff matrix conditions. But sometimes with increases in stiffness, you don't see any movement from the cytoplasm to the nucleus. And then in other cases, you actually see more YAP activity inside soft matrices than inside stiff matrices. And so a big question here is, how is dimensionality of the substrate actually integrating with ECM mechanics to influence cell state? And that's something that me and my collaborator, Padmini Rangamani, who's in mechanical engineering at UCSD, had fun thinking about. And we're both chemical engineers by background. And so we came up with the idea, why don't we treat the cell like a bioreactor, put components of this YAP-TAZ pathway where they should go. And in other words, some are membrane bound, some are cytoplasmic, some are in the nucleus or on the nuclear envelope. And let's see if we can answer the question of how dimensionality changes this signaling cascade and ultimately YAP-TAZ localization. And this really, is a spatial modeling problem. We need to think about the cell as a three-dimensional object. We need to think about it as having surfaces and volumes, and again, localizing components of this cascade to those surfaces and volumes. And so we defined a 3D cell geometry. We used a V-cell, virtual cell, for this project. And we wrote equations of change for each of the species in this cascade. And we wrote them in terms of cytoplasmic compartments and plasma membrane compartments. And as you're all very familiar, these are equations of change where you're just relating accumulation to what comes in, what goes out, what's produced, and what's consumed. So it's very fundamental engineering concepts. And then we took these equations and parameterized them based on defined experimental data. So for example, the phosphorylation of FAC due to stiffness has a range, and we would choose this range by mapping our model to the experimental data. So you can see we have good fits of our model to various components of the experimental data. And now, and this was all from 2D data. And so now we can start to ask questions of the model, like what happens when you change stiffness from the bottom of the cell to all sides of the cell.
Can I ask a question, Stephanie?
Yeah.
Yeah. This is Dennis. Yeah. Can you go back to your cartoon?
Yeah.
I just happened a couple of days ago to see a paper. I know the YAP task field is massive, right? Just drawing and of course predates the Piccolo work as well. So there's a lot of simplification, no doubt, going on here. But since you're particularly interested in mechanosensitivity, the paper I saw that appeared in Nature Com, I guess, within the last week, talked about important IMPORTIN, right? Getting into the nucleus through a mechanosensitive important molecule. Important 7, I think it was.
Yeah.
So in some sense, maybe effectively you have something, you're modeling it or not with entry through the nuclear pores. I'm not sure, but that of course is a cytoplasmic, nuclear shuttle, and it brings YAP more specifically, I think, in and at the same time binding to this particularly important 7. Its levels are mechanosensitive, but binding is competitive with other transcription factors. So of course, there's a lot more richness to this. But you have any comments on that? How getting into the nucleus, which is sort of all important here, has many aspects to it, right? And that are also mechanosensitive beyond FAC. And anyways.
Certainly, yeah. This, of course, every model is kind of somewhat of a generalization because we can't model all the components of the cell all at once, at least not yet.
And they're just still being discovered, right? So anyways, go ahead.
Discovered. Yes, but I will say that we have kind of two interesting, several interesting things going on in this model, specifically at the nucleus and where YAP is kind of integrating signals. So you can see that YAP is integrating signals from FAC, which is sensitive to stiffness, but then also YAP is integrating signals from the actin cytoskeleton. And the actin cytoskeleton, we have a component of the model that's acting from the cytoskeletal stiffness to change the nuclear stiffness that influences pore opening. So here we're just modeling like one pore, one type of pore. We're not like designating different ones, but we're saying that the cytoskeletal stiffness does influence the opening of these pores, and that influences transport across the nuclear membrane, along with phosphorylation and all of these other events. So certainly it's a generalized version, but this turned out to be very important. And having this component in the model also made the model sensitive to changes in nuclear shape.
So maybe that is equivalent to an import factor.
Yes.
That is expressed at higher levels on stiff than soft. Maybe there's some equivalence there. Okay.
Yes, I think that it's fair to say that idea is incorporated in here, if not explicitly.
Yeah. I'll send you the reference, but it seemed yet another new factor, mechanosensitive factor, and had some interesting competition to it. Thanks. Sorry.
Yeah. No, great point. Just to reiterate that we do have this effect of like actin, myosin coming together to influence cytoskeletal stiffness, and then that's impacting the level of lamin AC. That's then impacting the ability of nuclear pores to be stretched and to be open. So that did turn out to be a key component of this model. And so again, just to tell you that we parameterize the model to experimental data from 2D studies that have looked at different levels of activation as a function of stiffness. And our model fit very nicely to most of those, except maybe myosin a little bit fit lower stiffnesses, but less higher stiffnesses. And so we went on to ask some questions of our model, which is what happens if we change stimulus from just a basal stiffness stimulus to a stimulus that is on the entire surface of the cell. And we kept the same geometry, which is actually more like a 2D cell geometry than a 3D cell geometry, but we'll get to that in a minute. So first thing you can see is we can step through parts of the signaling cascade, and already, from the beginning, we see that we can start to establish gradients depending on whether signals are coming from the bottom or coming from all sides. And of course, if you're activating more surface area, as in the 3D stimulus, you're going to get more overall activation as well. So you can see slight increases in PFAC. These are volume averages here I'm showing on the right. And so you get slight increases in 3D compared to 2D stimulus. And that increase, or that difference, is carried through to row A activation and also to myosin activation. But what gets interesting is that further down, when we get to the nucleus and to YAP, there's a bit of signal amplification that happens. And like I said before, this is actually, YAP is integrating this signal from phospho-FAC, from the substrate, but also integrating information from the cytoskeleton and its influence on the nucleus. And that has a tendency to amplify the differences between the 2D surface activation and the 3D surface activation. So we see more nuclear YAP-TAZ in response to stiffness in 3D than in 2D. The difference is larger at this part of the cascade than previous parts of the cascade. But as I mentioned, these cell shapes aren't necessarily realistic for what happens to a cell when you put it inside of a 3D matrix. And so one of the next things that we wanted to consider is, can we decouple some of these variables that change, when you put a cell in 3D or 2D. And so what we wanted to do was, increase substrate activation area, but keep cell volume constant. And that has the effect of changing the shape of the cell. But also we wanted to consider different activation schemes and reaction area schemes. So here I'm showing you a 2D reaction scheme where you have activation area only on the bottom of the entire surface. And you can also restrict reaction area only to the bottom, which is for some reason the way that we intuitively thought about this problem in the beginning, but it turns out that this 2.xd condition is the more realistic case, where you have activation area on the bottom, but reaction can happen everywhere on the surface of the cell, and reactants can diffuse everywhere. And then you have this 3D condition where you're activating the entire surface and reactions are able to happen on the entire surface. And so if we take these different geometries of activation and reaction, and we increase substrate activation area, so that's these pink colors here, in each different geometry, we're going to have slightly different cell shapes for the same activation area. So for example, in 3D, to have a activation area of 200 microns squared, the cell is going to be rounded, and we're going to have a surface area of 200 microns squared and a reaction area of 200 microns squared. As we increase the surface area of activation, the cell surface area has to increase and the cell has to spread, but we're keeping volume constant. So that changes cell shape. So you can see how for different geometries, the same surface activation and the same volume yield different cell shapes. That's the main takeaway. So when we ran our model on these systems, and like I said, the 2.XD system turned out to be the most relevant to the experimental results that folks see. And actually there's data out there showing that, reaction happens everywhere on the membrane, even if activation only happens on the bottom. So in this type of geometry, if you increase substrate activation area, keeping volume constant, you have the cell stretching and becoming thinner and thinner. And you can see that as stiffness increases, which is this upward direction, you get increasing YAPTAZ localization to the nucleus. And then the more stretched out and flat the cell is, the more YAPTAZ localizes in response to stiffness. So that stretching out of the cell is actually amplifying the response to stiffness of YAPTAZ. But if we look at a cell of the same volume with the same surface activation, but in a 3D geometry where the activation area is spread out across the entire cell, we get very different cell shapes, right? And so if we increase, at the same surface activation area of 200 microns squared, if we increase substrate stiffness, we don't really see YAPTAZ responding to that stiffness. There's about the same amount of YAPTAZ in the nucleus across these different stiffnesses. But as we increase the surface area of activation and the cell has to spread out in this, but it's still being activated on all surfaces, the response to stiffness is then visible, where you can see YAPTAZ is starting to localize more to the nucleus in response to stiffness. So what this really is saying is that high substrate stiffness alone is not sufficient to generate a response in YAP nuclear translocation. Low stiffnesses are fundamentally robust states where changes in cell shape, substrate dimensionality, and nuclear shape do not affect outcomes. And I didn't show you nuclear shape, but you can certainly read about it in the paper. Similar kinds of effects. And interestingly, a rounded cell shape was another robust state, which was relatively insensitive to changes in stiffness. And so stiffness sensing through YAP is really tightly regulated by surface to volume relationships. The cell surface area available for reaction is a dominant factor. And nuclear shape, which I didn't show you, but it's also influences YAP localization. And then it's important to note that YAP localization is integrating both cell shape and substrate presentation information along with stiffness. And so in some cases, stiffness matters more, in other cases, cell shape matters more and dominates.
I have a question. Sorry. You said we could interrupt. So Dennis, again, yeah, can you go back to your data there? I know you do a lot of experiments, so I'm just kind of curious, like the upper left, right, with highly stiff 7 gigapascal, that's easily done with patterns on rigid substrates, going back decades, right? Mm-hmm. I just haven't kept up on whether, even DuPont did that. I suppose he did. I suppose they Piccolo and DuPont probably have that. Yeah, they had pattern substrates which were probably rigid, right?
Yeah.
Yeah. Okay. So so that probably fits, right? One should realize that state is also going back decades, low proliferation, right? So so one thought about that is, it's sort of out of the cell cycle. But actually my main question is, experimentally, how do you achieve that lower right where it's soft but spread? Yeah.
So I think it's informative to go back to this first figure where we had experimental data that was contradictory. And so I think this case on the right is probably what you're getting at, where, and I forget exactly what material this was, but the cells, and it's a function of cross-linking, I think, of this particular ECM condition where cells are able to spread out. So maybe the pore sizes are larger, but it's softer and they see more YAP activity. But you can see in the cytoplasmic stain in the stiff condition, the cells are more rounded and you get, I guess, perhaps more confined, and you get lower YAP activation. So I think there's a good match up with these experimental results here and what the model is predicting.
Okay, so there's something funny, odd about the matrix used, but this you think provides evidence of a well-spread cell on a soft substrate.
Yeah, inside of a soft substrate. Yeah, so I think
inside 3D.
Yeah. So in the next part of my talk, I'm going to talk more about confinement and degradability. And I think these are key parameters in 3D culture that tend to impact how cells sense the mechanics. So if you allow a cell to spread out because it's, less confining or more degradable, then perhaps it's able to sense the mechanics more effectively than a cell that has to remain rounded because it's inside of a highly cross-linked matrix.
Okay, thanks.
Yeah. Any other questions before I move to the next part? Okay, great. Okay, so
Stephanie, maybe one question.
Sure.
I don't know if you've covered this already. The cell sizes are also different on different stiffness. Is this incorporated in the model somehow in the way you parameterize the 3D shape?
We didn't change volume. We always held volume constant, but volume will definitely change the signaling for sure. Because the main message of the model is that the surface area to volume relationships are really what's the most important. So yeah, I know on 2D, for example, cells, pump out water and tend to lower their volume as they spread. That's not something we incorporated directly, but it's certainly something that would change the outcomes in the model.
Thank you.
Yeah. Okay, so in this part, I really want to talk about, what is a relevant in vitro context to study metastatic migration? And can we take what we learned from this, modeling study and kind of incorporate it into our model system to better reflect, the tumor microenvironment? So as I mentioned, we're interested in studying metastatic migration, and a lot of work has been done in the past couple of years, especially by Andy Ewald's group, showing that collectively invasive cells are the ones that are typically seeding or successfully seeding sites of metastasis. So we're very interested in this collective invasion phenotype. And our goal is to really figure out how to provide cancer cells with cues that will put them in this collective migration phenotype, but also in a relevant transcriptional state that mimics the tumor. And so obviously cell shape and surface activation area are key determinants, and 3D culture is probably necessary to achieve the same types of surface activation that are achieved in vivo, which is also 3D. But ECM type matters as well. There's plenty of studies, on the biochemistry of the material showing that it matters. And collagen type 1 is, of course, a major structural and adhesive component of tumors and has been shown to influence progression to metastasis. So our approach here was to try and engineer collagen to systematically change cell shape and thereby achieve distinct transcriptional states and migration behaviors, and then match those to the most clinically relevant behaviors and cell states. So the first thing that we did was we wanted to know, could we tune a collagen 1 gel and change its architecture just by using molecular crowding techniques. So here I'm showing you, the typical process by which we take collagen monomers that are dissolved in acid, neutralize the pH. Fibrils are nucleated and then grow, and this forms a collagen matrix with fibers that look something like this. And this is a 2.5 mg per mil matrix, which I'll refer to as P0 from here on out, or low density. And then we asked, can we put in some PEG, just some small 8 kilodalton PEG to induce faster nucleation rates to change the architecture of the collagen? And will that be able to influence cell shape and cell phenotype? And so, for example, when you study the nucleation and polymerization of the collagen matrix with just absorbance measurements, you can see that the P0 condition, or the low density condition, without any PEG, nucleates and forms a gel slower than if you incorporate 10 mg per mil of PEG. So P10 means 10 mg per mil of PEG. And then at the end of this process, we try and wash the PEG out to make sure it's not influencing cell behavior. So by titrating PEG, we were able to, just during the nucleation phase and the gelation phase, we were able to change the architecture of the collagen. And interestingly, this did not significantly change the stiffness of the collagen matrix, either by bulk shear rheometry or by local AFM tapping, which we did with Adam. Thanks, Adam. But it did tend to change the degradation rate of the collagen. So the degradation rate was lowered for the higher crowded or faster nucleated matrices. So next we put cells inside of these matrices and we asked, what shape does a cell take on? And we saw that in the matrices with longer fibers that didn't have any crowding, the cells were more elongated. And as the fibers got shorter, the cells got more round. And also this correlates with the degradation. So as fiber length gets shorter, you get more circular cells. And as matrix degradability goes down, you get more circular cells. And so we next asked, what does this lead to in terms of migration behavior? And so over the course of 7 days, you can see that cells that started out as elongated primarily migrated as single cells, and they're kind of spread out throughout the matrices. But with more and more confining matrices, meaning shorter fibers, less degradable, we see that the cells start to behave more and more collectively. And they go from this kind of chain forming pearls on a string kind of phenotype to something that looks almost fused in this P10 condition. And just to ensure that PEG wasn't doing something funny here, we did a control where we made a regular low density gel, let it fully polymerize with cells inside, and then added 10 mg per mil of PEG on top and left the cells for 7 days. And the cells still behaved independently. So it's not something funny going on with excess PEG being left over in the matrix. And so here I'm just showing you that as you increase the confinement, you start to see multicellular chain formation, and then you also start to see this kind of fused structure formation. And again, this is interesting because you can, with the same density of collagen, achieve very different behaviors. And then also, if you compare a high density collagen matrix to this low density crowded matrix, you can get the same effect because you have very similar architectures. And so you can look at the stiffness here and see that, in 2 mg per mil that has the same stiffness as the P10 condition, you get very different cell behaviors. But in matrices that have two very different stiffnesses, you can get the same behaviors. And so it's just showing that the confinement and the architecture of the matrix seems to matter quite a bit for this collective migration phenotype. And it's not just breast cancer cells that do this. Many different cancer cell types respond to confining collagen conditions this way. And even tumor explants from patients respond in this way as well.
Sorry, I'm sure you said this. The PEG was washed away from that. Is that right?
Yes.
No residual PEG, you said. I see.
Yes. But just in case there's some that we couldn't get rid of, we did this control experiment where we left the PEG on intentionally.
Uh-huh.
And it didn't cause any collective behavior.
Okay. If you didn't want, is that quite the same as not washing away the PEG? I mean, if you didn't wash away the PEG, you pull water out of the cell and this sort of thing. Is that basically the same thing? You put it on top. Okay, I guess.
Well, in these conditions up here, we tried our best to wash away as much PEG as possible. So we did many washes over the course of a day.
Yeah.
But just to make sure, because it's probably not 100% effective, right? So just to make sure that PEG wasn't causing somehow the collective behavior that we were seeing, we actually added PEG on top of a low density matrix.
Yeah.
And we didn't see
but
any
I'm just saying,
color.
if you didn't wash it away at all, then you'd have this hyperosmotic. I know Sean Sun, who's here, I think, right, has studied, that'll pull water out of the cell, right? And so would that have an effect? It also raised the question, what is the colloidal osmotic pressure, even with the PEG gone, of these gels?
Mhm.
That would I mean, because the assembly state is different, I guess the monomers are all, but the different gel states are hydrated differently, I would think, because they have different structures. So is there still an osmotic effect? And I guess I would ask, do you see changes in cell volume? Have you tried to measure that? It's tough, but
We've tried, and then we didn't feel confident in the
Sure.
measurements,
yeah,
so we haven't
I
reported
would
it.
feel I would feel the same. What I'm, because I have a bit of a blood cell background. If I threw a red cell into this, I would use that. That's an easier shape, and it's a very good osmometer.
Mm.
So okay, you're using an osmolyte to change collagen structure, and
Mhm.
you wash away the osmolyte best you can. That's all fine, right?
Yeah.
But in changing collagen structure, do you also change effectively the osmotic activity with hydrogels? It's a hard concept for, I don't know.
Yeah.
It's
It could be.
hydration. How much does the gel want to hold on to that water and pull water out of the cell?
Right. Well, and in higher density, you have less space for water anyway.
Yeah.
So yeah, it's definitely, there's probably something going on there, but I don't know what it is exactly. We haven't
Sorry.
measured it anyway.
Yeah.
Yeah, so we see this behavior happening with many different tumor cell types. And so we were interested to try and understand whether there was some kind of a conserved program going on here, or whether this is just like jamming or physically keeping cells in the same place without actually changing their transcriptional program or cell state. And so we asked, are cells changing their gene expression program? And are cancer cells doing it in a way that's different from other normal cells that undergo migration and collagen, so fibroblasts? And we compared genes that were upregulated in a high density collagen to genes that are expressed in a low density collagen. And so what we found is that there's this intersection here between two very different tumor cell types, and excluding anything that's normally upregulated. And there's 70 genes that are upregulated in high density. And these 70 genes, if you look at what they're enriched for, you see regulation of cell migration, anatomical structure development, multicellular processes, and especially blood vessel development and vascular development. And so a lot of these genes that were in these two categories actually overlap significantly with genes that have been annotated for vascular mimicry, which is a phenotype that happens in vivo in humans and in tumors in humans. That is a process where cancer cells actually form vessel-like structures that conduct blood flow. So this was really interesting, and it does suggest that this is not just a, networks forming just because you're keeping cells all stuck in one place. You're not just physically confining them, you're actually changing their cell state, their transcriptional programs. And lastly, to answer the question of whether this is really relevant to cancer metastasis, we took the 70 gene module and compared it to tumor transcriptomes from the TCGA. And we found that this 70 gene transcriptional program was predictive of poor patient prognosis, especially in triple negative breast cancers, in breast cancer, and also in 8 other tumor types. And we also found evidence of it at the protein level in breast tumors that also seem to have vascular mimicry going on. So I guess in summary of the entirety of part 1 of my talk, dimensionality seems to be able to override stiffness in some cases, and cell shape and surface area to volume relationships are really dominant especially in 3D. And confinement of cells, which is really a function of matrix architecture and degradability, is a key factor that's driving individual versus collective cell behaviors. And the collagen-induced network phenotype is clinically relevant to 9 different tumor types. And the utility of this is that predicting prognosis, especially in tumors like triple negative breast cancer, could help assist in choosing to take chemo or not. This is a big question for these non-targeted therapy, these tumors that have no options of targeted therapy. Do you want to take chemo and risk, heart failure and all the other things that can come with it if you don't know if your tumor is going to progress? And we're continuing to do functional studies of these gene modules in the hopes of identifying targets for selectively stopping this collective invasion process, or selectively killing the cells that are undergoing the collective invasion process. Are there any questions before I move to part 2?
Hi, Stephanie. This is Anurag from Texas A&M. Just a quick question. In your collagen-induced network model, have you looked at the localization of YAPTAZ?
We did not look at it in that model, no. Turns out we were thinking about these things very differently, but in the end, they're rather synergistic. So we should go back and look at that.
Okay.
I have a question, how does the migration phenotype enter into the picture of this YAPTAZ nucleus internalization? You see that stiffness sensing is enhanced by surface to volume ratios. So you will think that mesenchymal type cells will have higher YAPTAZ nucleus internalization.
Yeah. And so that's a good question. And there'll be more data in part 2 that will kind of address that. But these invasive cells that are more elongated and end up forming these collective networks, they do end up proliferating more, whereas cells that stay more rounded for longer periods of time downregulate proliferation. That will be coming up in a few slides.
Thanks.
Yeah. Okay. So I'll go on to part 2. So in this part, I really want to focus on the challenge of understanding migration heterogeneity among cells of the same type. So I showed you before that these MDA and B231 cells, if you put them in high density collagen, My computer froze. You can see that some cells after 24 hours will elongate, divide, and start invading. And then other cells will kind of stay rounded and undergo this rotational migration behavior. And then those cells will form these spheroid structures. And so we have about 86% networks, and 14% of these spheroids are acini. And so what is it that's causing these cells to choose one way or the other? And also we see this not just in the MDAs, but several of these other tumor cell lines as well. So to figure this out, it's actually pretty challenging because, here you have very few of these non-invasive spheroid structures, and to try and selectively isolate those guys is challenging. And we thought about just doing single cell sequencing and hoping that clustering algorithms would be able to tell us which cells belong to which phenotype, but I was very skeptical about that. And so we actually developed a way to physically sort out these phenotypes and then do sequencing analysis and try and understand what's the difference between these. And so to do this, we expressed Dendra2, which is a photoconvertible protein, in the MDA and B231s. And we seeded them again as single cells in the high density matrix. And then over 7 days, they form these clonal structures, which are either non-invasive or invasive networks. And then we came through with a laser and a galvo scanner to photoconvert selective types of structures. So for example, in one round, we'd photoconvert the spheroids, and then we'd digest the matrix and sort them out. And then in another round, we'd come and photoconvert the networks and digest the matrix and sort them out. And so what this looks like is something like this, where beforehand, all the cells are expressing green Dendra2 and no red. And if you come through and choose a structure like this spheroid here and photoconvert it, you can turn it red selectively. And then when you go to flow sort it, you can pull those red cells out. Another cool thing that we're still exploring is the use of this to actually track individual cells within structures. So this is a really fun tool to use. But the first thing we did was, in being able to sort out these different populations, we wanted to understand whether they were plastic or static in their phenotype. So we sorted out networks and we sorted out spheroids, and then we reseeded only the network cells. And we found out that if you reseed networks, they stay pretty much in this invasive network phenotype. But if you reseed the spheroids, they mostly produce networks, and only a small but larger percentage than the parental population produce spheroids. So this suggests that the non-invasive spheroid phenotype retains some plasticity and can transition into the invasive collective migration phenotype. And it also suggests that the regulation of these two phenotypes is at the epigenetic and transcriptional level. We were also curious if either of these phenotypes correlated with stemness. So we stained for CD44 and CD24. And we found that both networks and spheroids were comprised of CD44 positive, CD24 negative stem-like cells. But interestingly, some of the spheroids actually were CD24 positive. So we're still kind of thinking about what that means, but it's just interesting to know that there's not, sorting phenotypes doesn't sort stemness necessarily. So the next thing we wanted to do was sequence these phenotypically sorted populations. And again, the reason is that we really felt like understanding the transcriptional programs for each of these phenotypes was important. And we didn't trust that just doing single cell sequencing without phenotypic guidance would give us the right answer. And it turns out we were right. So if we just look at the single cell sequencing results from an unsupervised perspective, where we're just asking algorithms to cluster cells based on how similar their transcriptional programs are, we see that we get 2 clusters. But if we overlay our knowledge based on sorting of the networks and the spheroids, you see that the networks are actually, sparsely distributed in this larger cluster, and then the spheroids are in both clusters. So this unsupervised clustering algorithm really does not capture migration phenotype differences very well. And if we look at the differentially expressed genes that we would have gotten from assuming, these 2 clusters are of interest, we would have found these genes. But then when we compared differential expression between the actual phenotypes, we found this other set of genes, and only 108 genes overlapped between these 2 analysis methods. And then 70 genes were completely unique to the phenotypically sorted analysis method. So I think this just goes to show that, within a cell type, it's really important to guide this type of analysis and not rely on unsupervised methods. When we went in to look at what are these genes representative of, what are the processes that are differentiating these 2 phenotypes, again, we saw different enrichment terms. And if we focus only on the phenotype guided enrichment terms, we found that the network cells are really biosynthetic, very metabolically active, very pro cell cycle, and also experiencing some oxidative stress and ER stress. Whereas in the network or the spheroid phenotype, we find more like immune signatures and negative regulation of cell proliferation. So this was really kind of surprising, but also interesting. So we wanted to validate whether these enrichment programs and genes that we discovered with phenotypically guided sequencing were functional to the actual phenotypes. So first, we kind of wanted to check whether there was a difference in proliferation, because we saw that the spheroid cells had strong negative regulation signature. So we stained for Ki67, and indeed we did see that the network cells had more Ki67 staining. And turns out they're more susceptible to paclitaxel as well. The next thing we checked was this immune system signature that was kind of surprising. The spheroids had upregulated several immune system modulators, and in particular, several HLAs. So we stained for HLA-A, and interestingly, in the network phenotype, the HLA-A was kind of localized maybe to the perinuclear ER, but not really to the plasma membrane. But in the spheroid phenotype, the HLA-A was primarily localized to the plasma membrane. And so this, along with several other genes in the signature, suggests that the spheroid phenotype likely would be more interactive with the immune system than the network phenotype, which might be in kind of stealth mode, if you will. And next, we targeted some of the genes that we found differentially expressed to figure out if they were actionable targets. So we selected heat shock protein 90, which was upregulated in the networks. It's also upregulated in numerous solid tumors, and it correlates with unfavorable prognosis. It's a molecular chaperone, and it's thought to induce angiogenesis and promote metastasis. And its ATPase domain can be inhibited by radicicol. So we tried a treatment with radicicol. We also targeted DEK, which is highly expressed in the network phenotype as well. It's an epigenetic and transcriptional regulator and can be inhibited by cordycepin. And then lastly, F3 was upregulated in the networks, and it's a gene encoding tissue factor, and it's overexpressed in breast cancer tissues and thought to be a mediator of progression. So in this case, we actually added recombinant F3 to the medium. So inhibiting These top two is expected to reduce or limit the invasive phenotype, and then adding F3 is expected to enhance the invasive phenotype. And indeed, that is what we saw. If we tracked early invasion, we saw that radicicol and cordycepin reduced invasion, but F3 increased invasion. And at day 7, treatment with radicicol pretty much prevented like any structures from forming. Everything remains kind of as a single cell. Cordycepin reduced the number of networks that we found, and recombinant F3 didn't seem to change the overall number of networks, but it did help in the early invasion. So in summary for this part, phenotypically supervised single cell sequencing is really able to provide improved selectivity for identifying differentially expressed genes. And it provides, pretty functional hits within these phenotypically guided analyses. And information about cellular process coordination also came out. So for example, we selected based on migration, but found strong changes in immune signatures. And ultimately, the collective invasive state was highly biosynthetic, proliferative, and perhaps immune evasive, although we haven't confirmed that, and more susceptible to paclitaxel, whereas the non-invasive cell state was more homeostatic, anti-proliferative, and immune stimulatory, and less susceptible to paclitaxel. In ongoing work, which we're hoping to publish soon, we're really trying to figure out, is the cell intrinsic or cell ECM interaction driven? So we're doing single cell measurements for multiple cell ECM interactions simultaneously to understand these initial conditions that drive these divergent phenotypes and transcriptional states. So in response to your earlier question about Hippo pathway, the cells that remain rounded ultimately ended up in this state, which was anti-proliferative. And the cells that were able to elongate early on ended up forming these collectively invasive states, and they were more proliferative. So that does suggest some differences in potentially in Hippo signaling. And so with that, I have to thank all of the students that did the hard work. Kirsten Scott was a big part of the modeling effort, along with Dr. Padmini Rangamani. And then Kevin Chin did the single cell sequencing and sorting experiments. And then Will, Saral, and Daniel were the ones that did a lot of the matrix engineering and migration analysis. And then Hannah Carter, our collaborator, who helps us do a lot of the bioinformatics analyses, is at UCSD, as well as Adam, who helped us do some of the AFM experiments. And with that, I will take any questions.
Thank you. Fantastic work. Any questions?
I have a quick question. So yeah, fantastic talk. I was wondering if you had thought about expanding your simulations from part 1 to sort of capture multicellular or the collective migration that you showed in part 2.
Yes, that is a very natural next step. And it's an important caveat to note in the first model, we don't have any feedback from like LATs and cell-cell interactions. So yeah, good point.
So the HSP90 inhibitor, we usually think of HSP90, I think AB1 also as fairly constitutive and one of the most abundant proteins. So yeah, it usually associates with growth in the number of cells. It didn't surprise you, that this just wiped out the cells there?
Well, I guess we were thinking that we would still get the spheroid phenotype, but we didn't get that. We just got single cells. The spheroid phenotype is more proliferation limited, but so it basically proliferates for a few days and then stops. And so I guess we could have treated later and probably seen the difference we were expecting, but we treated it kind of immediately.
Yeah, no, it should really shut down growth because all these proteins need that ATP dependent folding. Yeah, even at 37 degrees, it's not a heat shock. It's a constitutive chaperone, very abundant.
Yeah, yeah. But we did see differential expression. That's why we chose that one. And perhaps it was
Yeah,
too much of a hammer.
Yeah, but
treatment.
did the differential expression, there's AA1 and AB1, but I think I remember AB1 is the constitutive. So, but does that just correlate with mitosis?
I don't think so, because we did see other heat shock proteins and like stress granule proteins, which we're following up on. In general, it seems like, actually, if you zoom out, it really seems like both phenotypes are kind of different responses to stress. And I think in the culturing cells in high density collagen is a stress, much like hypoxia is a stress on the cell. We see indications of that. I didn't present it today, but we do see indications of that and changes in the metabolism early on of cells when you put them in there. Their glucose uptake is limited, and you see kind of redox stress type reactions changing. And so I think the high density condition is a stressful condition, and these two phenotypes are two different ways of dealing with it.
Thanks.
And there are some other folks that I talk with on campus that try and understand, how hypoxia and other types of stresses can converge on similar molecular programs and behaviors. And they are of the belief that the stressed state is the metastatic state. It's almost like cells get out of there because they're stressed. And it may not matter necessarily which stress you apply. It's just enough of a stress that they leave the situation.
Great.
Any more questions? If not, let's thank the speaker one more time. Next month, we will continue our journey in San Diego, and we will hear from Adam Engler. So yeah, take care and see you next month. And thank Stephanie for your great talk.
Thank you so much. I enjoyed the discussion.
Yeah.
Bye-bye.
Bye.